Our technology platform is a combination of enzymes we've discovered and developed new pathways, new strains, new fermentation strains, as well as access through our collaborative partners like the USDA that has one of the largest strain collections available. We have access to that, special access to that, and world-class advisors, and then our know-how as well too. So, okay. So what I'd like to talk about first is what we've done in the xylitol area. Polyols are specialty sweeteners. They're used in certain products like gum and candy and other products, confectionery products. And sorbitol is by far the most dominant product out there. It's cheap to make, made from glucose, and it's very inexpensive. But products like xylitol and mannitol are a couple polyols that have some very unusual and useful health benefits. Xylitol in particular, xylitol is thought to actually help prevent dental caries because of the way it interacts with the bacteria in your teeth. And so actually it's the #1 sweetener in gums and a number of other confectionery products in Asia, and it's growing in Europe. In North America it's pretty much non-existent because of two reasons. One, the supply stream to make it is limited, and it's a little bit expensive for products in the US and Canada and North America. So really, if you could take xylitol and make it the main sweetener in sugar-free gums, you could significantly impact health of the gum. So in order to do that, though, you have to solve the issue with supply. Right now, xylitol is primarily made by the chemical hydrogenation of xylose, which has been purified from the paper and pulp industry, specifically from birch trees. And I believe all the world's supply of xylose from birch tree paper and pulp is used and converted to xylitol. Actually, if you look at the growth of the market, xylitol went from $20 million a year to $150 million a year in 1 or 2 years, and then it stopped growing. It grows very slowly now, and the reason for that is you just can't get the raw material. You have to start out with a very pure xylose, and if you don't have pure xylose, if there's other sugars in that mix, then you get a mixture of polyols which are difficult or impossible to separate, which completely changes the properties of the sweetener. So what we've done is to put together a process that can overcome the two main issues. One is the feedstock supply, and two is the cost. Okay, well, I just talked about this. But again, the current chemical process is this. You need purified xylose, which is expensive. It's actually cheap to run the chemical hydrogenation, and then if you don't have pure, you need extensive purification. And whereas our xylitol bioprocess can start with hemicellulose, which is one of the fractions of agricultural and forestry products. And it's inexpensive in abundant supply, and we can start with a mixed C5 and C6, 5-carbon and 6-carbon sugars, which is very important. We don't need pure xylose, which is a 5-carbon sugar. We can have a whole series of sugars mixed in there, and our fermentation organism just either consumes the sugars that we don't want or converts them to xylitol.